Hi, everybody. It's been a while since I've done one of these, and sorry for the delay, but life happens. So today I'm going to give you another one of these updates from Science and Nature, but this time instead of it being the diversity edition, it's going to be the science edition, and it covers work I've been thinking about in May. As before, I've taken several articles from the readings of Science and Nature and I've made some little commentaries for you, and I've shared them on with you because I find them interesting, and I hope that you will as well. The first article that I'm going to show to you is this one, and it's an article. It's a brief news article in Nature. I've got the URL for it at the bottom of this PowerPoint, and hopefully you could stop screen and see it. And it's called Gene Transfer from Plant to Insect Spotted. And this is about it's a short article about what I'm calling the plant-insect eternal struggle. In this eternal struggle, insects eat plants, then plants make compounds to poison insects, so the plants don't want to be eaten, and so they try to stop the insects from eating them. And then the insects evolve ways to resist the compounds. In this case, we're looking at a pair of white flies, and this is here. And this white flies are important because they are, among other things, they're a pest on your house plants. If you live in the north and try to grow plants indoors in the winter, they are a huge damage to agricultural crops, and they can act as a vector for over 100 pathogenic plant viruses. So obviously people interested in agriculture want to find ways, figure out how plants themselves can repel white flies, and it would be better to have the plant do the poisoning than for us to spray insecticides on them. So in this case, research scientists discovered that some of the white flies that they were studying have gotten a gene directly from the plant. And it acts inside the white fly body to neutralize plant poisons. And these are sometimes called phenolic glucosides. So the phenolic compounds in plants are often what make a plant poisonous or toxic or distasteful, even to humans. So what they found was that if a scientist inhibits this particular gene inside the fly, it dies when it dines out on plants with phenolic glucosides. But if the gene is acting normally, it manages to disable the glucosides from the plant. On further examination, it turns out that this gene actually was originally a plant gene, and that at some point it got transferred to the fly genome. And this is pretty interesting because gene transfer is very common in microorganisms, but less common and less well known for transfer of certainly across the kingdoms, like a plant to an insect. It's an important finding because it offers a pathway for lessening white fly damage, and it offers really interesting insights into the mechanisms of predator prey evolution. And it's the first confirmed example of a natural gene transfer from a plant to an insect. So that's plants. The rest of what I'm going to talk to you about has to do with animal embryos, and specifically with human embryos, because there's suddenly a whole group of papers about the study of early human embryology that are of considerable interest. Oops, wrong way. So why am I starting with this book? Oop, come back. This book, which is a book called The Dialectic of Sex: The Case for Feminist Revolution by Shulamith Firestone. Starting, and it was published in 1970, and it was a revolutionary book when it was started, when it was published. And it has in it the claim that the only way women can ever be liberated is if the gestation of embryos is taken out of the body and done in the test tube. And in my consciousness raising group in 1970 with a group of other young feminists, we discussed this book. And I thought, gee, what a cool idea. We just do the baby making in the test tubes. And then nobody's burdened by having their body taken over to make a baby. Many of my friends who were also my age were scandalized by the idea. I remember this because I was so surprised, because it seemed to my little scientist head like a perfectly reasonable approach. At any rate, it seemed also pretty science fiction-y in 1970, and it still is, but things are happening that may make it someday a possibility. So I'm going to report to you on 3 articles, and here are the 3 links to them from Nature and Science magazine. And they are in the March 19th and March 19th edition of Science and the March 25th edition of Nature. And the report on new research featuring lab-grown structures that mimic the early stages of human embryos. So Shulamith Firestone, who is no longer alive, but she will be laughing in her grave, I do believe. So here are the 3 articles. The first page of the 3 articles, and I'm going to tell you a little bit about them. They are all about very similar kinds of things. In this article, it says key human embryo state. I'm sorry, that's this one. Lab-grown structures mimic human embryos' earliest stage yet. In this one, it says modeling human blastocysts by reprogramming fibroblasts into eye blastoids. And in this one, key human embryo stage mimicked in the lab. And in the original article, it also has this same graphic of a human embryo. So what's going on with these? Let me tell you a little bit about them. Let's see what else I'm looking at here. I'll tell you a little bit about them in the next. So what's happened is that multiple research groups have independently report that they have grown balls of cells that look like human blastocysts. This is the first stage or almost the first stage of embryo development following fertilization. And blastocysts normally form about 4 days after an egg is fertilized by a sperm. And these experiments offer a window into a crucial time in human development and an opportunity to better understand pregnancy loss, of which more in a minute, and infertility without experimenting on human embryos. So to the authors of this is a way of studying early embryogenesis while avoiding the ethical dilemma of experimenting on human embryos. They don't have the feminist take on it that I offered you in the very beginning, but that's why I told you there's another way of looking at these things. Some of what scientists currently understand about this early stage of development has come from studies on human embryos, but access to these embryos is limited and it's tightly regulated because of ethical considerations. But the blastocysts grown in the laboratory come from human stem cells instead of human embryos, and so the hope is that they might avoid some of the ethical limits on human embryo research and could increase access to this type of work. They do not expect the new blastocyst-like structures to have the ability to grow into a complete embryo. And previously, this kind of work has been done successfully on mouse embryos, but now, for the first time, several labs have done it on human embryos. So let's look at this a little bit more. Let's explore a few of these points. First of all, what is a blastocyst? Second, where did these stem cells come from? And third, what are the ethical limits and who has set them? So what is a blastocyst? The human embryo is normally fertilized shortly after it's ovulated out of the ovary. So it bursts out of the ovary, it's out in outer space for a minute or two, that's a metaphor in that case, not a precise measure of time. And then it's swept up by the ends of the oviduct into the upper reaches of the oviduct. Right in this period, right up here in this early part of the oviduct, it's fertilized. So sperm, meantime, have swum all the way up, and they not only swim, but they're propelled by the contractions of the uterus and the oviduct itself. So it's a mutual process that gets the sperm up here. And then they fertilize the single egg, and the egg, the single cell egg, and the fertilized egg, which is now officially a human embryo, begins to move down the oviduct. It moves down, as it moves down the oviduct, the cells start to divide, first 1 to 2, then 2 to 4, 4 to 6, you get the idea. And 4 to 8, 8 to 16. And by the time it reaches about here in the oviduct, it's become a solid ball of cells called a morula. And that's after 3 days after fertilization. And it's at this point, it has 16 cells in it. It continues to move down. And as it moves down, what happens is that the morula begins to differentiate into what's called a blastocyst. These cells, the inner cells in the morula, start to secrete a fluid which pushes some of the cells out to the outside, forming an epithelium around the embryo, and a group of cells stays in the middle, to one side, and that's called the inner cell mass, and that's where the embryo itself comes from. And then there's this fluid space in between. The epithelial cells ultimately become part of the implantation process and ultimately join up to become part of the placenta. So implantation then starts to occur in the upper reaches of the uterus around 6 to 7 days, and the implanted embryo begins to really burrow into the uterus, into the uterine wall by 9 to 12 days, and begins to form into a 3-layered embryo by a process called gastrulation by about 12 days. So what they're doing is trying to mimic this blastocyst stage. They're getting cells to form a blastocyst, which is a group of cells that look like the inner cell mass, have fluid separating them. And what they're doing is using stem cells to do this. The stem cells, and let me see if I can magnify this for you for a bit. So the stem cells, I don't know how to move this out of the way, but the stem cells, oops, let me get this out of the way, can have two and depending on which lab was doing the experiments, they used 2 different possibilities to get their stem cells. One group took stem cells from human blastocysts. Where did these blastocysts come from? Why? They were leftover embryos from reproductive technology facilities in which people had donated eggs, or they might come from a spontaneously aborted early embryo. Anyway, they dissociate them and use them to form human pluripotent, that is, with many potencies, stem cells. Or they took adult human cells and they treated them with a bunch of chemicals, growth factors, that made them revert down to this early undeveloped stage, and then plated them out. And again, they became human pluripotent stem cells. They put them in a special kind of agar plate. They cultured them first here, and then they put them on a flat plate and helped them, in a 3-dimensional plate that allowed these cells to aggregate into what they called a blastoid, which looks a lot like a human blastocyst. There's an inner cell mass here surrounded by an epithelium. Let me get this out here for a minute. Here. So, as I said, they used this single step 3D culture process to generate the blastoids. There we go. All right, now moving on to the next slide. The question though remains is that there are ethical issues about stem cell research, and these have been even about stem cell research. And I think in a future YouTube, I'll talk a little bit more about those because the rules for these have just been changed. The Trump administration forbade all of this early stem cell research. The Biden administration has just revoked the Trump administration's rules. We'll take a look at that. But what I want you to see here is that there are ethical research. See, this comes from an NIH publication, and the ethical issues are different at different phases of stem cell research. So first is the donation of biological materials. In the previous thing I said, they either got the cells from leftover eggs or from spontaneous abortions or skin cells from adults. And in any of those cases, they had to have gotten informed and voluntary consent. And by voluntary, I mean, you couldn't say to a woman who was having a spontaneous abortion, well, we'll do the D&C and help you heal, but you have to give us the embryo to use. I mean, that had to be completely independent of treatment. So the research with these cannot involve the destruction of embryos, the creation of embryos specifically for research purposes, the payment to oocyte donors. They cannot involve medical risks. The ethical issues involved include what are the medical risks of oocyte retrieval? How do we protect the reproductive interests of women in infertility treatment, et cetera? So there's a whole bunch of these that go into consideration in anybody doing one of these studies. But what these experiments offer is a window onto a crucial time in human development and an opportunity to better understand pregnancy loss and infertility without experimenting on humans. And I, of course, offer the futuristic feminist critique, the futuristic feminist vision that maybe someday babies can be gestated in the test tube. That would involve a lot, including inventing a placenta that worked and a whole bunch of other things. So I don't think it's going to happen in my lifetime, but it's something that some people do have an eye on. And the feminist question of how would that liberate women if they didn't have to actually be the ones who gave birth to children. But anyway, the question of offering a window into a crucial time in human development, and an opportunity to better understand pregnancy loss and infertility, leads us to the next story that caught my eye. And that is this article, where again, I've given you the URL, and it's called, the title of it is, it's a tricky title, but Cooperative Epithelial Phagocytosis Enables Error Correction in the Early Embryo. And let me take you through this a little bit so you understand what this means. First point is that there are loads of errors in the early embryo. That is, that when I taught embryology to pre-med students, I always noted that at least 10% of all fertilizations never make it past first base. That is, the embryo dies usually pretty early. Usually there's a spontaneous abortion, spontaneous fetal loss, and more rarely the embryo or fetus survives. But there are defects of various sorts. They can be minor. or they can be severely disabling, or they can even lead to death of the fetus at a much later stage. Biologically, the effect on the woman of losing a blastocyst or a morula is much less, although it can be significant, than losing a fetus that's developed for 3 or 4 months. So, the process of fertilization and producing a viable and successfully developed baby 9 months later is fraught with biological errors, and they happen a lot. So, so these folks are studying some of the ways in which some of the mechanisms embryos have evolved to correct errors early on. So it's not just the miracle that there's this flawless development, but mechanisms have evolved in which the embryo can correct early errors and proceed normally. So, as I've said, it's such a complicated process, yet in the end, wonderful little creatures, a wonderful little creature is born. How can that be? Well, not, as I say, not so much. Nature, as we're calling her here, makes a lot of mistakes. She isn't quite as amazing as she seems. 10% of all fertilizations not make it past the first 13 weeks. And these fertilizations are often not even known. They result in spontaneous miscarriage. And by the way, if abortion after 30 days becomes illegal, as some states are now moving towards, and if the Supreme Court upholds this, women will be prosecuted for natural developmental errors. And there'll be no way to tell the difference, and it's punitive against women having bodies that make babies. Okay, so let's look at this though. What the heck is phagocytosis, and what is epithelial phagocytosis? And let's take a look at that. We start again with the sperm developing the egg. This is now outside of the context I originally showed you, but remember, this is happening in the upper reaches of the oviduct. And as it moves to the middle regions, you have this totipotent morula stage, this solid cell ball, and then it moves to this blastula stage, in which you have the inner cell mast cells, which are the cells which go on to form the embryo proper. This whole thing forms the human fetus and the placenta, the embryo component of the placenta, and these inner cell mast cells. Inner mast cells are pluripotent that means they can form in the end all the tissues in the body, or most of the tissues. So in this though, the epithelial cells here, which end up not forming part of the embryo proper, but form the various membranes that surround and protect the fetus. These epithelial cells are capable of undergoing phagocytosis. That is when one cell engulfs another and digests it and destroys it. So the epithelial cells are capable at times of recognizing malformed cells with chromosomal abnormalities, and sort of dropping down and engulfing it. So what happens if one of these inner cell mass cells dies? As I said, some do. You have this, and this is kind of a big blown up. You have what's called epithelial phagocytosis. And it's cooperative. That's what this paper has discovered, that is, several cells will work together to snag an embryo. And here you see that the epithelial cells have receptors that bind this dead cell, and as it begins to bind it, the epithelial cell grows up and begins to cover and engulf the dead cell, and then the membranes up here fuse it, and this dead cell becomes internal. And there are a couple of different cells involved. There are some cells that sort of reach down into the inner cell mass from the epithelium and help move them up to the surface, and then there are others that do the engulfing. So several cells work together to snag the cell here. to engulf it, and then once inside the cell, they digest it. And thus rid it from the developing embryo, enabling what's left in the embryo to replace the dead cell and to continue to develop, hopefully successfully. So, recap. We start off with a one-off. We started off in this YouTube presentation on the ever-evolving relationship between plants and their predators. The twist here is that a white fly managed to obtain a gene from a plant that helped it resist the poisonous effects of a set of chemicals in the arsenal. So lesson 1 from that article is that evolution is a wonder to watch, and it's a very powerful process. Lesson 2 is the next time a scientist promises you that a field test of, this is still about the white flies, promises you that the field test of an organism with an altered genome is safe, and that there is absolutely no danger of the gene escaping into the wild. This is the response I think you should give them. They could be right, but don't count on it. And in terms of recap for embryos in Petri dishes, these are some of the questions to ask. Does this put us on the path to test tube babies? Does using stem cells get around the ethical issues involved with direct experimentation on early human embryos? And so many people have been able to have children using assisted reproduction, but what happens to all these embryos? And then lastly, development is risky. A lot of fertilized eggs die, miscarry because one of the hundreds of thousands of chemical or mechanical processes involved in producing a successful, healthy fetus goes wrong, is mistimed, etc. But mammalian development has evolved some mechanisms for timely correction. Phagocytosis of dead cells is one of these, and several epithelial cells may cooperate in the process. And I'm going to stop the share and let you know that this is the end of this presentation on the science edition of What's New in Science and Nature. And in a few weeks, I'll be back with some more articles from Science and Nature, items that I think are socially important and items that I think are just biologically interesting and fun. And I hope you've enjoyed these, and until next time.